So it's really with a big pleasure that I introduce Professor David Canella from here from ULB that will present us something.
Yes, thank you very much for the nice presentation. Well, first things first. Warning, that's a talk with a high content of cross-disciplinary information and so on. So you're going to be out of your comfort zone. So the comfort zone is a risk. Go take an extra coffee if you want. Yes, I'm going to bridge something on smart agriculture, biorefinery, all those concepts that, are kind of, up in the air, and try to bridge it. And so to give you some focus on how one can help the other. To do that, I have to take it a very long way from, the car crisis that we have now in Europe. And then I have been told that I have 2 hours, right? No. How much I had? 45? 30? 40? 40, okay. I'll try to make a good use out of it. The last program that, the very long title that nobody remembers of the BrIAS, the first edition was Past, Present, and Future on Food Production, Agriculture, Sustainability, and Climate Changes. There you are. Maybe I have either lost or added one word, maybe. So, good. So let's say, I told you, I have to start it from a long distance. So you know these numbers. So these are the numbers of cars that we have in Europe. 250 million cars, more or less. Look at this other number here. I know that many of you are engineers, so I will flex my muscles. Ready for the battle. This is the max production that we achieved in the last 3 years per year of electrical vehicle. Around 2 million electrical vehicles per year in Europe. You having only Western, let's say, technology, not counting the Chinese, let's say, production system, or let's say that this car were also having some Chinese, let's say, or Asian, let's say, train production. So let's say some parts were done with Asian parts, but because European banned the batteries that comes from, let's say, Asian countries, let's say that these numbers, so the 2 million per year, are generally made with parts that come from the Western production. Do you think it's okay? I mean, look at the rate of production. And this is the max we can do, the best we can do. 2 million per year, and we have to replace 256 million of cars. It will take more than 100 years, unless something changes. So, I mean, when people talk about the future of electric vehicle and so on, the future is already not born yet. I mean, like, the future is not even a reality. I mean, not even today we can talk about this as a reality. So we will have a big problem if you don't tackle this issue of sustainability in car in, let's say, in a serious way. This is not serious. I mean, going around and say that the future is electric vehicles when we can produce only 2 million per year, forget about it, okay? So something has to drastically change. I don't know if we have to change the way we produce batteries, the way we produce things, but let's say that that's not the future. So what about the other 250 million of cars that are still running with thermal engines? They are pretty much divided like this. So 70 millions of tons of fuel is considered for gasoline car, then under 80 millions of diesel car. Millions of tons. So if you go back to the original number, 250 millions of car, you might think that each car consumes sort of a ton of gas or fuel. Mm? But although we have done a lot of policies against diesel, you see diesel is still the one that takes the majority of the whole, let's say, fuel consumption in Europe. This is all European numbers. Obviously because we have these heavy trucks that still runs on diesel. Road transportation for goods is the major one in Europe still. So let's say that although the private-owned cars are moving towards, or let's say phasing off diesel slowly, we still have a huge proportion of diesel consumption. Remember, these 70 million tons of gasoline will go back to that, this number. So now the numbers are there. So now I made up the numbers. So we have a lot of cars to replace, maybe not. We have a lot of thermal engines still, and we will not be able to replace all these thermal engines in due time to meet the requirement of CO2 reduction, for sure. And we are still consuming a lot of gas and fuel and diesel. When we look at where this energy comes from, you see that 60% of this energy, and we'll say like petroleum products, and so this 250 millions of, let's say, tons of fuel is actually here, is inside there. So that counts for the energy that comes from outside Europe. So we have the two first line, natural gas and total petroleum products, that they all come from outside Europe. And that makes up like 60% of the total energy consumed in Europe today comes from outside. Then you have the one that nobody wants to, let's say, still use, and we should actually ban it. That is the coal. That is the last one. The solid phase fuel. So this is the last part of the, let's say, the energy, so the reminiscence from the past that we should actually phase out. And so the only two things that remain, let's say, interesting for European policies are the two in the middle, the renewable energy and nuclear energy. Those are the only two kind of technology, a source of energy, which are scalable in Europe, which we can make it in an independent way, and so make it, let's say, in a way that we could use this one to replace the 60% that come from outside. Now, what's the problem with these two kind of, let's say, source of energy? One is that the nuclear energy will provide you mostly electricity. And so for the electricity, you need like a lot of electrical devices to consume it. And I don't know if we are there yet. Again, the problem of the electricity that you have to consume it, and it's very hard to store it. So the stocking of the electricity is, again, a problem due for batteries. So now in the field of renewable energy, or let's say the new energy, let's say it, now they came out with this new way of thinking, this power to X, using electricity to convert this energy into a form of energy that is stable, that you can store it. Usually it's a liquid, an organic carbon-made form of energy. So we'll come back to this one. So this slide was just to show you that the only two realistic solutions that we have in Europe are either nuclear or the renewable energy. When we speak about the renewable energy, we have all these 6 major sources. And all of them, so hydropower, solar, geothermal, wind energy, marine energy, they have in common that they are more or less, let's say, developed. We know wind energy and solar energy is much more advanced than marine energy. They all have in common one thing. They need a grid. They need a network. The electricity that is produced, or even the hot water that comes from the pipe of the geothermal, has to be distributed, so they need a network. Either if it is cable, or if it is like a grid, or let's say a pipe, still have to be produced. Again, we have this problem of stocking the energy. The only one, again, that can solve this problem, it is the bioenergy. The bioenergy that is one of the renewable energy, that is the only one that can provide you with a form of energy that is either in gas or in liquid. So when we speak about bioenergy, or let's say bio-e-fuels, we speak about converting biomass into either biomethane, that is now a very solid reality in Europe. We are actually doing very well in that. Or biodiesel, colza, palm oil, and so on, or bioethanol. And bioethanol comes from actually the sugar that comes from the plant. Right. So this is the old generation. When we speak about synthetic fuels, the classical fish and troughs, let's say, mechanism, designed during the First or Second World War, remember, taking carbon monoxide and hydrogenating this one to form, let's say, methanol and so on, but with a very high temperature. So this is, let's say, very inefficient at the level of energy. But it was good to create liquid fuels for the German troops when they had to fight the war. They were like under the embargo of, let's say, petroleum. So it's like one of those very emergency sources of energy. So forget about this, but don't confuse when people speak about synthetic fuels, they refer to this. So if they put the E in front, this is the new kind of E, or synthetic fuel, that is actually taking electricity instead of energy, temperature, to do a similar process, starting from CO2, but not the CO, so the CO2 that is atmospheric, hydrogenating it and taking the hydrogen because of the electrolysis of water using the electricity that is produced in a green way as a wind power and so on, and then produce again methane or methanol. So this is the future. So this is going to be the new or the new synthetic fuels at the E stage for the electricity that is being used for this process. What we have in the middle is this today. So today we are here in this, let's say, amount of energy that we can produce in a renewable way, bioenergy, that is liquid or gas, and this is a reality. I know that many people don't realize that we have a lot of technology already developed, but it's not so popular yet. So, but this is totally scalable. It's already like a demonstration plant. I will now guide you through all these, let's say, examples of these biorefineries that are, let's say, the base of what I was doing during my PhD and my early research career. So I'm really spinning out from this kind of research. And so I wanted to show you that when we talk about biorefinery and, let's say, lignocellulosic ethanol, or let's say second generation, third generation of biofuels, we talk about not anymore using something that human can consume as a food. So we are not talking anymore about potatoes or rice or sugarcane or let's say palm oil. We are talking about something that is only dedicated for the production of fuels and does not compete with food. That's super important. People still remember the infamous biofuels because they were produced, they still produced today, starting from food. But the new technology, the new generation of biofuels are all coming from, let's say, processes that do not touch any edible biomass. What we're talking about, mostly lignocellulose, so the part of the plant that we cannot digest as a human, because we don't have the enzymes that digest this particular part of the plant. So in French, we say la tige, in English, the stem, okay? So the woody part. The third generation that is mostly dedicated for the production of biodiesels, that is instead coming from algae. So these are all photobioreactors installation where you grow your algae that accumulates lipid, you open the algae cells and you take the lipid out, and these lipids are exactly the same kind of calorific power of diesel. And you can use this one to replace the diesel. And instead the ethanol and all the alcohols that we produce in the lignocellulosic degradation and then transformation into biofuels are there to replace the gasoline, okay? So what we are targeting when we do the processing of lignocellulose, we are targeting the cellulose inside the wood. So remember when you were in high school, so we all studied how the wood was made. Remember there was like the cell wall that is sort of, created by this very stiff and let's say polymer that is called cellulose. Some of you today still have some cotton in your t-shirt. I know that today is still like 80–90% polyesters, but the lucky of you that still have a cotton t-shirt might feel it. So that is the cotton, and that is made of cellulose. Then in a natural way, you have the hemicellulose that coat all around. And so this is what we call it hemicellulose or pectin marmalade sort of saying. And then you have the third component that is the lignin. So for decades, we studied the way to unlock the treasure of the sugar, so the cellulose, to penetrate there and then take this reservoir of cellulose and to expose it, so to produce the glucose out of it. Glucose because at the end we will ferment this glucose in forms of ethanol. Likewise, the beer process. So it's pretty much brewing. The only difference is that in brewing, you start from the malt, that is very easy to obtain the sugar, the glucose. Here, we have to win the millions of years that nature spent in protecting this cellulose, so to make the plant more resistant, so that we can have wood that stays there since thousands of years. In some of these archaeological wood is still untouched and not degraded by, let's say, bioaggressors. And so we can see that the whole nature, let's say, development have worked against us. So now we designed a fuel technology to get there and then to take this sugar. Why this so appealing? Because if we go at the numbers, a little bit of the numbers, I have to give it to you again. The whole production of this reservoir of sugar, of cellulose, lignocellulose, in Europe, considering only the cereals. I'm not talking about wood or other kind of, let's say, plants, just the waste of the agricultural system that we have in Europe. We are talking about 500 million tons of mass. 500, so this number here. We'll come back, no worry. So that number there. So the 442 million tons, but we can approximate around 500 million tons. Only considering cereals, or let's say agricultural waste. Well, by doing the calculation and considering the efficiency quite at the maximum, which is the state of the art of the today technology in the conversion of lignocellulose to final ethanol, we can obtain around 80 million tons of ethanol. That is equivalent to 60 million tons of gasoline. Only taking the full cereals waste of Europe, we can actually almost replace the entire gasoline consumption of Europe in form of ethanol. So you see now the appealing of this technology. It's there. That's why we consider the only scalable technology that we have in Europe if we want to replace tomorrow the gasoline instead and then using ethanol instead. So it's there. Now, how do we do it? It's a very simple process. Even master students now, they master it. So we offer a course here in ULB where we offer like 24 hours of a practical lab where they actually learn on how to do this process from A to Z. And so at the end, they go back home with a little bit of ethanol produced from either wheat straw or wood. So that's part of the course that we offer. And these are pictures from the students, actually. So this is like the wheat stem, like it is when you just harvest them. We actually do an hydrothermal pretreatment that is cooking. So cooking at like 100 degrees, no more, for a few minutes. Then we take the enzymes that are produced by fungi, which are our nanomachines. They are super fancy. They look like small dinosaurs with a cavity where they can extract like a single chain of cellulose and then chop the cellobiose glucose unit one after the other, walking through this, let's say, railway made of cellulose. And so these nanomachines that are developed by fungi are there. We usually walk in the forest to harvest new sources of enzymes, and then we simply produce this one using a little bit of fraction of the sugar that comes from the wheat straw. So again, the circularity theme is there. So we produce the enzyme by using some of the sugar, just like 5%, no more. And then we use this one to liquefy the wheat straw into a molasses that looks like that. The only difference between the picture on the left and the one on the right is that we added only enzyme. I mean, the water that you see on the right is the same amount of water that is on the left. It's only trapped by capillary forces, and so you cannot see it. But as soon as you digest the whole polymer, you actually liquefy it. Yeah, it looks like petroleum, but no, it's not. That is just a coincidence. What we want to do after that, it is a fermentation. So we simply add yeast, the one that we use for pizza at home, exactly the same. Then we obtain a transparent liquid that is the ethanol that is distilled out. So you have the bioethanol, which is not green, by the way, just transparent, let's say fuel, that is totally compatible with the new generation of engine. You simply put inside. Now, this one is sold as E85, ethanol 85% in few stations. In Europe, by law, so if you walk in any gas station, we don't have this E85, we only have, by law, a little fraction in each of our gasoline. It's like 7, 8, 9%, maximum 10%. So if you now see on your gas station, there's a number, there's a letter E, and then a number after. That number tells you what is the percentage of ethanol inside your gasoline. So only few gas stations in Europe now offer this 85, okay? But we are getting there. I want to just drag your attention, and this is the only chemical things that I will ask you to learn today, so it's the biochemical things that I will show you, how these enzymes work for degrading the cellulose, because it's a marvel, believe me. So these enzymes are just depolymerizing the whole cellulose by snipping this, let's say, let's call hair, or let's say chains of cellulose from the chain terminus. Let's say that they can recognize where the chains finish, so they enter, they penetrate, and then they start to chop after each other. And so they start to create this unit of cellobiose, they do unit of glucose, and then with this last enzyme, beta-glucosidases, it can cut down into glucose. That's not complete. You always need a gate opening, so an extra enzyme that creates the new chain termini, rupture into structure, so where the enzyme, the C-beach, the big enzyme can enter and destroy the whole cellulose. Those were the original cocktail enzymes that have been developed until 2010, but they were struggling delivering, let's say, a profitable technology. Let's say that usually we stopped at 60–70% of degradation of the entire, let's say, lignocellulose. Not good for making money. Until 2010, when a pioneer group of scientists discovered a new enzyme, just one single enzyme, which changes everything. So that is one of the moments where you just find one single enzyme that completely pivots the entire field. So far we always talked about hydrolysis, so that this cellulose was cut by hydrolysis, adding water to cut the bonds between the glucoses. But with the discovery of the LPMO, that is an enzyme that is able to oxidize the cellulose, we actually started to gain much more efficiency, because this enzyme is able to cut in some regions of the cellulose where the other couldn't, and so synergize much more, and then produce, let's say, more chain entry for the other enzymes to destroy. So once we started to add this enzyme, we started to observe more beneficial, let's say, and better efficiency, that if we report here in sort of, by time and then efficiency, you can see that in usually 4, 5, 6 days maximum. So if you see the first black line with the square, that is the, let's say, the old generation of the cocktail, stopping around 60%. Instead, with the new generation containing this new enzyme, we can get up to 85–90%. This is at suboptimal condition. At industrial condition, today we are talking about 95–97% of efficiency of conversion of the original biomass into final product, that is the ethanol. That is like usually a jaw-dropping, let's say, reaction that I have from, let's say, chemical engineering, because usually in chemical engineering you have, like, when speak about biomass, you are happy if you have 60–70% of efficiency, but 97% is super high. And so it became profitable. So we kind of surpassed the red line of profit. And so now they are actually running to create all this new demo plant. Don't get amazed by this picture that looks like, oh, fantastic, they are already like at the industrial level. These are just toys, okay? These are just small toys that, for example, this one has been built when I was doing my PhD in Denmark, in Copenhagen, nearby. Today is part of the Academy of Biomass Transformation that is like open there. So today it is a facility that students, new generation students, can go there to learn. It's not an industry at the level of scale and so on. So this is just like a toy. Very expensive toy, shall we say? But look at the numbers, what they can produce. 5,000 tons per year. We are way, way, way below the millions of tons needed. So let's say that they are not a real scale. So the real scale of this one should look like sort of 30, 40, 50 times bigger. And then if you have in mind what is a usual oil refinery as a size, I would say that this part here just occupy the parking lots for the personnel of a normal oil refinery. So it's very small. So, but these were instrumental to show that it works. And so now the good thing is that not only from cereals, so wheat straw, but you can see that many others started to use other kind of biomass. It's very interesting, the one in Estonia by Fimeron that is now using birch wood. And so also now the wood is entering the game. And so now we are unlocking another, let's say, box of treasure of lignocellulose that was not really tapped before. And then the one in Italy that struggled at the beginning, but now is catching up. Again, is taking marginal land, let's say marginal lignocellulose, the lignocellulose that is built in marginal land. In Italian we say canne, I don't know, like should be like giant reed in English. So those kind of, let's say, lignocellulose that comes from, let's say, marginal periphery of a cultivated land. So again, do not compete with first generation or let's say sugars and so on. Why we don't see them more? Of course, so the final price of this bioethanol, the E85, is below €1 actually, super attractive. The biodiesel the B95, it is a little bit higher than €1, 1.1, 1.2 usually. But we still don't see the many gas stations offering these products. I just zoom here on the region where we are living, the north of Europe. But I also saw a little bit in Germany, some of them, but they are not so popular. So there are maybe a few hundreds of these, let's say, gas stations. Reason why? CapEx cost, and then a very low oil price that is, quite of a big concurrence. Imagine that, this is the price today, but if you have to think of adding all these CapEx costs to produce this one, it has like a sort of inertia. So we will pay back, but the price that is now selling is too low. So the margin are very, very little. So nobody wants to put money from the pockets and then start to invest on this technology, having this little margin. So this is the major barrier for the biorefinery. And here comes the next phase. So trying to drag out of the fuel trap this technology. Because, in the meanwhile, we've been investigating all new source of technology. Believe me, like the way to unlock this lignocellulose with pretreatment, so the cooking, the enzymes technology, how to modify this biomass and so on. And then we just figured out that it was not only fuels possible to produce, but a full array of products that goes from biomass materials, advanced materials, drugs, and biochemicals, new chemicals, oligosaccharides, food replacement, prebiotics, and many, many, many more. So many that, at a certain point, there was a very good initiative that was already in 2005, if I'm not wrong, to regroup all these industries that works around the valorization of lignocellulose under a huge consortium that's called the bio-based industries, that today counts more than 300 or 400 established industries, not just spin-off, industries, okay, which are profitable in making product. Of course, then there is a lot of other spin-off. Each university today counts one small spin-off working on the valorization of biomass. That's the legacy we obtain from the journey of investigating how to make biofuels out of lignocellulose. And we have finally also a joint undertaking that is one of these, let's say, branch of the Horizon 2020, that is the Circular Bio-Based Europe, that is promoting and is offering a lot of, let's say, financement and calls and grants for, let's say, these thematics. So those are just small pictures for demonstrating you what we are doing today. So some of them comes from my lab and so on. So we learn how to make nanocrystals, nanofibers, so nano-sized, let's say, polymer out of this cellulose, controlling it with enzymes, of course. So you see, this is cellulose. Don't think that is a POMA. So this is just cellulose that is trapping water in a way that can become an hydrogel. So imagine that you can put your bioactive compound inside, that you can use this as a cream for delivering your drug to your skin. That's it. Transparent wood. Yes, we made it transparent without losing the, let's say, the polymer structure. So if you touch it, it's still, dense as wood, but transparent. And then we also learn how to use these new enzymes to crack in a specific way so that we can control actually the nano size, the size of the crystals that we are obtaining from the cellulose, again with enzymes. Instead of going to production of alcohol, we are actually now producing bioplastics, the PLA. Instead of making the alcoholic fermentation, just with a lactic acid fermentation, you can create the precursor for making the PLA. This is now produced in this way. And this is the last, let's say, revolution. So far I talk about only sugars, but don't forget that we also have lignin. They are the brown part of the wood that is today have been unlocked for producing all those new materials, advanced materials. So those are all these now common things that we can buy in any brico, which contain actually part of lignin injected, more than injected inside. Chemicals, as you can see, if you can manage to obtain the molecular, let's say, separation of these molecules, you can get up to 1,500 euro per ton. So it's quite, of a valuable, let's say, product. And finally, so this one brings me back to my last 10, 15 minutes, kind of. Yeah, 15 minutes of, let's say, applications that is in agricultural science. Because we just figure out that if we stop or we control the degradation of lignocellulose until a certain point, we do not go all the way to sugar, to the single monosaccharides, but we can stop until a certain point. That is the production of oligosaccharides. And so now the idea is to valorize these oligosaccharides in the way that can be like used as a prebiotics, or the latest, let's say, idea we got is to use it as a plant vaccine. So this is just like, take-home message. We can read it later on. I'm now excited to show you this plant vaccine application. I don't know if you heard about that, with these climate changes happening, we have more and more of these, let's say, wave of pathogens, microbial pathogen. I'm not talking about pests as an insect, but just fungal or microbial pathogen that attack the plants. So now we have today one of the most harsh to eradicate that is Botrytis cinerea. That is one of the most devastating pathogen that is causing more than 100 billion euro or dollars of worldwide, let's say, loss in equivalent in food. It can attack up to 500 plant species. So it's pretty much, there is no crop virtually that can be attacked by, that can't be attacked by botrytis. So it is one of the most important to fight against with. Today, the state of the art says that we are using pesticides, fungicide to tackle this problem. And so the idea is that, we should replace this fungicide with something else. These fungicides are usually very bad for our, let's say, body. Usually they even disrupt our hormone system, or they can even destroy completely the soil microbiome and render the soil pretty much almost, I won't say sterile, but very less biodiverse than it was before. And so the idea is to use something that is replacing it with, let's say, biologically derived molecule. The other biggest cons that we have with the fungicides is that they induce resistance. So year after year, you have less and less amount of little different kind of molecules that you can use to spread on the soil. But instead, the use of plant vaccine, it completely reverts the paradigm. We are not spraying against the fungi, we are spraying on the plant to teach the plant on how to protect the plant itself against the fungi. So it's just a way to awake, to stimulate the immunity, to tell the plant, hey, you are under attack, or you will be under attack soon. Raise your defenses, and then the pathogen will arrive in 1 or 2 days or 3 days. And so the plants are sort of, let's say, prepared for the fight. So this is the technology that we developed here at ULB, using these oligosaccharides that are, let's say, produced by the degradation of cellulose with these new enzymes that I just talked about. The first things we did is to test if it was working. And so indeed, yes, we saw that it was sort of protecting the plant up to big extent, 70%. So let's say that the spread of the disease on the plant was reduced of 70%, counter the plant that was not protected. So it has a quite of a promising effect. The good thing is that we learn all this by looking at the way the pathogen were attacking the plants. So we simply infected a plant one day, and then we noticed that by infecting this plant, this pathogen used the exact same enzymes that we used in the biorefinery for degrading cellulose. And then it took like, I want to be proud to say half an hour, and maybe 1 or 2 years to make the connection. That probably, let's say that, you know, the two things could have been very, very valuable, or could have been used for teaching the plant that could be relevant. So if you have, you know, the same enzyme that is attacking the cellulose when the cellulose is dead, but also when it's alive, probably is there where we should look at the source of this virulence factor from the point of view of the pathogen. So we started to study the biology of the pathogen. Again, cross-disciplinary. I dealt like with bioreactor design and so on, chemical engineering during my PhD, and then during the postdocs and the initial part of my career, we have been relearning all the biology since the fundamentals. So going back to the DNA, looking what is the most expressed gene during the pathogenic activity of these fungi, that was not easy. But we saw that, you know, this enzyme is fairly highly expressed during the pathogenic attack. So we isolated the most active. We use these enzymes to digest again the cellulose. We just grab all the oligosaccharides that were produced. And now we thought, okay, let's try to give these oligosaccharides to the plant to see if the plant recognizes as a signaling molecule for rise the immune defenses. So we tried, and then we succeeded in a good way, sort of say. So we observed that, you know, there was a lot of, let's say, again, protection. So the one before was on tomato, this is on Taliana, and so we have, again, a lot of protection going on. That is not enough. So to have a technology that works is not enough sometimes to, let's say, use it. Sometimes you have to really understand very well how it works. And you start to discover new features, which are, I have time, fantastic. Because now I think this is the most interesting things for the engineering coming on now. So one of the things that you learn by doing things for the first time in a field that you enter, like, you know, from yesterday, is that many things are already done. But then you have to really force yourself to search for the things that was not done, that you spend more time in reading literature to see, okay, what is not done? And then you realize that it was like, you know, something that's very interesting, because you look in a way that the people of the field doesn't look the same things in the same way. You look as an engineering point of view, or let's say application point of view. And then we're like, okay, let's start to see if there is some molecules that we can use to crack the code. If this molecule can be used for, let's say, signaling the plant in a different or unusual way. And so we studied the way the fungus, let's say, attack the plant, and the way the signal is spread. So the first things that happen when the fungus arrive to the plant, it just trigger a very quick, let's say, signaling within the plant. And so this is a picture, not from my lab, of course, it's from Simon Gilroy from Wisconsin Madison. He did this experiment just by putting like a caterpillar on a leaf. Then in a few seconds, like 20, 30 seconds, there was like a sort of messenger being produced in the plant that spread through all the plant. You see, in 20, 30 seconds, you have the signal that travels through, let's say, the cytoplasm of all the cells of the plant that have been spread all around. So although the infection, sort of saying, the caterpillar started to injure the plant in one side, on the other side of the plant, the leaves were already ready to rise the immunity defenses. This changed everything. So imagine that if you can touch a part of a plant and then the signal reverberate through the rest of the plant, that is very important. So now this is what we are doing. We are treating like only the sum of few leaves of the plant, because we know that the entire plants can be then protected, or we are just simply treating the seeds, because we know that the mature plant that will come after the seeds being treated will carry on their defenses again. So this is a very important feature now. Few minutes later, so now we are away from these 20, 30 seconds, like a few minutes later, you have an entire metabolomic reprogramming of the cells. The cells understand that there is something wrong with the surface, and then start to produce, to change completely the metabolism. It goes under inflammation, so this is one of the typical effects that have been always observed with the immunity. So also the plant get inflammation, and then at the end, after a few hours, 1 or 2 hours, let's say, the plants start to produce volatile hormones to tell to very distant part of the plants that they are under attack. Sometimes let's say the internal message travels, but then it needs a second message later on that arrives from outside to tell to the plant, okay, we are under attack, I confirm it, rise up your, let's say, defenses even stronger. But this one goes outside the plant and is not only catched by the same plant, but also from neighboring plant. And now you start to understand that we can treat one plant and then influence the plant next to it. And this is not only observed with ethylene, that is this very small hormone, but also from other kind of gases, let's say, molecule. So this nitric oxide does the same. And the important thing is that, you know, when you have multiple of these molecules arriving from different corner to a plant that was not attacked, it's just like rising more and more the defenses, and so it just will sort of create like a more complete shield for the next pathogen attack. So you have one plant that received the pathogen, it can, by bombarding of hormones, the neighboring plants, it can actually induce a full protection. The research on this regard is now wild. So we are now looking for all those molecules that can be, let's say, exchanged from plant to plant. So the very fast one are ethylene and nitric oxide, but now let's say this is a very small molecule, but now there are even specific metabolites that takes days to be produced. And so those one are usually associated with more, let's say, immune reprogramming at different stage. Of course, you know, each plant has its own specificity. Each pathogen has its own, let's say, kind of aggression mode. So probably all this diversity on different kind of molecules that can be exchanged from one to another plant is not just like a universal language, it's actually different dialect that the plants speak each other. Okay, so you should see in this way. Then there is this very old theory, and that was already formulated in the 87 when they could not completely, not even understand this old part because they had no molecular insights within this, let's say, theory or dynamics. So actually all those exchange of hormones is today, we are recognized today as at the base of this push and pull theory, where you can pretty much grow your plant in an intercropped way, where you can put like some plants that are very good in repelling the pest and others which are able to, let's say, attract them. Probably because they are exchanging the right, let's say, hormones that push the pest to fly away and some other which are able to attract. So now, learning how to intercrop and how to, let's say, crack this code is super important. The last 2 or 3 important, let's say, basic information is that now we call this one cry for help, let's say, style. So we not only search on the aerial part, but also in the soil. So the same things happen in the soil, where the plants is actually producing an exudate that can recruit, let's say, beneficial bacteria. So this is, again, very interesting from the point of view of smart agriculture. So to enhance the property of the soil. And the last discovery, that was a couple of years ago, is that the plant emits sounds, of course. Yes, they emit sounds, but we are not able to hear it. They can travel up to 5 meters away. And so when the plants are injured or stressed, they actually start to emit sounds to signals from each other. And so the neighboring plants can hear about it. And so the discovery has been done, thanks now to the engineering aspect of the entire field, because somebody have created like let's say, a microphone device, or let's say an acoustic device, very precise, on the very low, I think it's infra, yes, it's on the very low, let's say, range of sounds that human ear cannot hear. So you need to modify the microphone or the device. and also to create a model to recognize these sounds and to associate if it was like a sound produced for a drought stress or a pathogenic stress and so on. So now this model is now, let's say, just at this first example, is now going wild because each plant has its own kind of frequency and a kind of, let's say, sounds that can produce. And so this is now the new future trends in the next decade that will be developed. So not only hormones as a sense, so we not only need sensor for the hormones to understand what the plants are talking about, but also what they are really exchanging as a sounds. And so by being able to crack like this important, let's say, code, we'll be able to understand how the plants communicate among each other. Yeah. So just to go back to some few final slides, and to conclude the stories on how we discovered all these things from tomato actually. So it's that we gave these sugars and then we start to observe some reprogramming of the plant. We look at the DNA reprogramming, a lot of different, let's say, responses. The first thing that we observe is that the plants start to produce the fungicide. This is the biggest, let's say, sign of success because we told the plants, hey, you're under attack. These are the part of your body that is, let's say, being liberated from the pathogen, kind of. And then the plant itself, even if there was no pathogen there, started to produce the fungicide. And so in case the pathogen could arrive, it would have found the whole surface of the cell covered by this fungicide. So a very hostile, let's say, field to invade. We confirmed that by chance, and this is now not anymore a chance, but now it's a reality, this whole technology works without any inflammation. So for the first time, we are observing a new signaling that does not induce the plants to go through inflammation. We are not yet understood why, so we're doing a lot of research on that. But we like this because, without inflammation, the plants are not stopping the growth, so they keep growing and they go actually even faster because they're not dealing with something that they have to cure themselves. Yeah. Instead, they produce ethylene instead of the ROS signaling. And so we observe that even distal plants can be alerted. The plants are usually also protected for drought stress, so it's the same signal. So this is quite interesting for you to understand that, different signals can give you different kind, the same signal can give you different kind of, protection, not only by pathogen, but also from abiotic stresses. So these are the plants that have been treated that are much more resistant than the one untreated. They survive like 45 degrees in the oven, actually. Yes, and then just wanted to show you a few, let's say, picture of how they looks like the tomato. So we've done a huge campaign of experiment where we treated the plants with a several amount of these sugars at different frequencies and so on. And so we noticed that the plant started to grow actually and started to grow flowers and flower much earlier, like 2-3 weeks earlier. Since because they were not dealing with any inflammation, they could actually grow faster. Fruiting as well, so they started to advance the fruiting phase much earlier, 2-3 weeks. If you're not thinking about, you are in a greenhouse and you can anticipate the production of your fruits by 2-3 weeks, so cutting down 20-30% of your time, so that's a lot of money that you can save. This is the way the plants look like. So the one on the left is always the one never treated, and so the more and more treatments, you can see that the plants go higher, that they mature the fruits earlier. So those sugars are all kind of, model sugars that we have in our blend of sugars that we produce. The amount of fresh weight of harvest, so is also much higher. So we don't only protect plants, but we actually also stimulating the plants to produce more fruits. And so usually we have like up to 20, 30, sometimes even 50% more fruits by weight. And this is the way it looks like. So this is a classical plant that's been treated, and that one it was not treated and harvested at the same time. So as you can see, you have many more mature fruits. And so as a number of, let's say, per gram of plants, we have up to 11, 12 grams per plant. That is like, for these particular plants, represent 20-30% more fruits. So yeah, it is a sort of a payback So you should not even convince the farmer to buy these things because he's actually paying back itself. So the more money they make it, they can pay back the protectant molecule. Now it's over. So this was my last slide. As you can see, I try to bridge from going to a problem of substituting fuels with a technology, and this technology by chance also gave us new other kind of technology that now we are using for the application in agriculture. So we need a lot of cross-disciplinary, let's say, skills on how to apply these molecules. So the engineering aspect here is very much understudied, so it needs to be catched up. So these are some of the pictures of the people that have been working in this project. So the very early part of my group, and now the second part. I want to also thanks a lot Christian that is down there. Has been my kind of senpai or a senior colleague that have been driving me through this first 7 years here at ULB. So many thanks, and then I'm open for questions if you have.
We have a first question.
Thank you for this nice presentation. So you mentioned that these biofuels you are discussing are produced from non-food plants, but still there is a huge need of land to cultivate the birches or the whatever plants are used. And so isn't there a competition for land which could occur even though you don't use food plants?
No, because you are taking the plants residual of the food production. So you're simply living on top of the amount of plants already cultivated for your own primary product. That is food, or let's say timber for the building of a building and so on for this let's say sector. So you're only using the part that they do not use. So you're actually not increasing the demand of land for this. Of course, if then there is something profitable, then they will push for having more and more land cultivated for. But there is a lot of land available today used for the meat production. That is a response to the name of soja, soya cultivation. That is only proteins produced for cattle, sort of saying. And so if there is a little bit of land to take, I would say the competition should go to that direction. So to reduce the meat producers. Yeah.
Other questions?
What's the catch on the vaccine plus bio booster? Because it sounds too good to be true. And I guess the question then would be, why hasn't the plant naturally evolved to do that on its own instead of being told to create that fungicide?
Because the fungi, they can avoid it. So actually the fungi, they can create countermeasures to shut down the immune responses of the plant. So if you just watch the two having like a fight, you will see that they have like numerous actually sort of signaling molecules that can one impede the other. And they are always at the minimum amount of concentration just to overcome a little bit the defenses or the attack of the other. They are never at that big amount they can just cancel the fight and say, hey, now you raise the defenses. So this is what we can do as a human, control the amount of sugars that we can give it, like way more that is in a natural condition. Why? Because these are harmless for the environment. They are biocompatible. You can even eat. They are edible, these sugars. And so they are not causing any resistance to the fungi. That is the contrary of the pesticide, where you have to be really careful how much you're giving and so on for not encountering these whole problems. So, yes, it's too good to be true. It's true, but it actually works. So, and then we have the first generation of this is already actually on the market. It responds to the name of pectin and chitin and chitosan. So now they're already being sold a lot. I would say that virtually all the 99% of production of organic food is done by that, is sustained by these things. But there's only two products, with many more. So now this is the first that comes with cellulose, which is super abundant. The next will be the one based on hemicellulose, and then will be the one based on lignin, and so on. So the more of this biopolymer we can, let's say, unlock, the more, let's say, arrows we'll have to shoot back the pathogens.
Thank you.
Okay.
Thanks, David, for an amazing presentation. Now, from a chemical viewpoint, you talk about an efficiency of 97%. I guess that is the azeotrope between the ethanol and the water, no?
The what?
The azeotrope. I mean, at some point, the distillation of ethanol with respect to water,
Yeah.
you cannot continue distillating. That is why you have 97%. You can add, in this industry, they do add additive to improve that, because I know, for instance,
Yes, that's why it's
in
called
distilleries,
yes,
they are
yes,
moving
yes.
that azeotrope towards.
That's why it's not 100% ethanol, it's 85%, because you have to add an additive to reach to, let's say, so 15% is blended.
Perfect. And the second question is also about the efficiency, no? We're talking about 97%, but it's the transformation of biomass towards ethanol. But what about of the efficiency of each
Carbon.
joule produced with respect to each joule consumed? Because in this case, we have to cool down. We need a lot of enzymes, as you said,
You're
fermentation.
If you ask this question, you're an engineer. So
Yeah.
you know that you cannot transform one joule to the same joule. I mean, you have to, otherwise you go against the physics law, the thermodynamic law, sorry. So of course you have to invest energy. And actually the price you pay for that, that's the price that costs. So you have to invest a little bit of energy to obtain more energy out. So, but this is what you are doing, is like you are transforming something that is solid in liquid. So that's what you are investing energy for. And then the majority of the loss in terms of efficiency is at the level of carbon, by the way. Because you start with a molecule that is a C6 carbon, you obtain the ethanol that is C2. So you obtain 2 molecules of that, so you have at the end, carbon, four carbons to be rescued. You started with 6. Actually, at the level of mass, you're losing sort of half of it. But let's say all these efficiencies calculated at the level of, let's say, amount of molecule that you can obtain. Yeah.
To be a bit polemic about that, if you have to pitch in front of investors for this process, and they tell you, what about biomass gasification? If you have biomass and you use it for ethanol, or you can use it to burn it because it's more efficient from an energy viewpoint, and you produce hydrogen. So what is the catch, as Marco said, to sell this technique or this process, let's say?
Likely, I don't have to deal with investors, so it's not my job. Our job is to provide, let's say, technology. My smartest reply to this question is always one, and so it's actually the dogma of the biorefinery. Never design a process that's only giving you one single, let's say, avenue of how to valorize your product into only one thing. Actually, biorefinery should be a collection of different kinds of processes that allows you to diversify your products and to actually adjust to the market requests. Now, because of the oil price going so much down, so there is no point of producing something that is competing with oil. And so could be that producing gases would be much more, let's say, profitable. But then you have to design your biorefinery to be able yourself to also go their way. So yes, the answer would be to have like the real biorefinery should have all of those, let's say, avenue possible, and so producing a range of different products: gas, liquid, biomaterial, biochemicals, not the single product. This will never fly. You're right. Yes.
Okay, so I have a last question, and then we will go for a coffee because we need it.
Yeah, no.
And the question is the following: in some of these communication mechanisms between plants, is there any case in which plants that get a signal retransmit the same signal?
This is going to be the next paper. Not that I've seen it, actually. So normally the plants, all those signals are only for the one I know, but maybe somebody can know more regarding these topics. So usually they just prime the plant. So they just alert the plant to say, hey, you're about to be, let's say, under attack. But of course, if then there will be a pathogen, they come and then attack the plant, now the plant will be ready to respond, and within the arsenal of responses, it will also again reproduce the same molecule that has been used for triggering, priming the defenses, that's for sure. But now, the amplitude of the signal, is it the same or will be different because it has been primed and then attacked, responded, you won the battles, is the same or not? Is the same kind of combination of different kind of hormones, sounds, or let's say exudate, or is different because this is sort of second generation now, because you have the first one that has been just been attacked and then send the molecule, but the second generation is the one that has been primed and then attacked. Who knows? This is a very good question, actually.
So with this, I'd like to thank again our speaker,